forget to subscribe my channel and you can share to your friends as well. In today's video, we are going to look at another type of immune cells. These are granulocytes. These cells which contains granules in their cytoplasm. They include neutrophils, eosinophils, basophils and mast cells. How these cells developed and how they are involved in generating immune responses. So without further ado, let's get started. Now, what are granulocytes? Granulocytes mean cells that contain granules, as its name indicates itself that granulo means granules and cytes means cells. So these are the cells that contain granules in their cytoplasm. So granulocytes are the cells which are characterized by the presence of specific granules in their cytoplasm. There are basically three types of granulocytes that work together to get rid from body's infections or pathogens, and each type of granulocytes has specific granules having their own combinations of chemicals and enzymes. So you can see in this picture, this is granulocytes. Okay, this is neutrophils and this is eosinophils. You can see there are small spherical sac-like structures that are present inside the cytoplasm of these granulocytes. These are granules. Sometimes their number of the granules are so much, or they are in numerous amounts, so they can conceal even the nuclei or the nucleus of the cell, like that of agranulocytes, which are also known as mononucleate cells. Granulocytes are known as polymorphonuclear leukocytes, abbreviated as PMN, PML or PMNL, as they are having varying morphology of the nucleus that make them different from agranulocytes, which are known as mononucleated cells. So granulocytes are known as polymorphonuclear leukocytes or WBC, whereas agranulocytes are known as mononucleated cells, because in granulocytes the nucleus that you can see is of varying shape. They may be bilobed, trilobed or multilobed, but in the case of agranulocytes the nucleus is having a similar kind of structure, okay, slightly notched or spherical, but in granulocytes they are varying in the morphology of the nuclei or the nucleus. Now, a condition that is known as granulocytosis. This is a condition where number of the granulocytes increases, that indicates an infection or an autoimmune disease or blood cancer. There is another type of case which is known as granulocytopenia, where number of granulocytes in blood decreases. It may result due to congenital or acquired defective formation of granulocytes precursors. That's why the number of the granulocytes formation is decreases abruptly, and that causes granulocytopenia. So there are two different cases. The first one is granulocytosis, and where the number of the granulocytes increases, and the other one is granulocytopenia, where the number of the formation of the granulocytes decreases. Types of granulocytes. Generally, we say that there are basically three types of granulocytes, which are basophils, eosinophils and neutrophils. But mast cells are also comes under this category. These all cells are characterized by the presence and the types of granules they are having, which also distinguish their work and the shape of their nucleus. Let's learn about these granulocytes one by one, but before that, let's make our understanding better that how they are formed. Now, the formation of granulocytes. So we have just seen that from the hematopoietic stem cells, two pathways has generated. The first one is myeloid and the other one is lymphoid. From the myeloid, we have seen that monocytes has formed, and in this slide, we are going to look how the granulocytes are formed. So first, in myeloid stem cells, with the influence of interleukin-3, interleukin-5 and granulocyte colony stimulating factors. With the influence of these factors, the myeloid stem cell, or we can say the myeloid progenitor cell, get differentiated into myeloblast. Okay, then this myeloblast generates three types of promyelocytes. The first one is basophilic promyelocytes, the second one is eosinophilic promyelocyte, and the third one is neutrophilic promyelocyte. So we can see from the myeloblast there is a generation of promyelocytes. Okay. Now this cell is blue in color. Why this is blue in color? Because the granules that are present in the basophilic promyelocytes stained the methylene blue. That's why they appears blue in color. Whereas in eosinophilic, the granules stain red. Thereby these are red in color. Whereas in neutrophilic, the cell looks quite midway between these two blue and red color. That's why it appears quite pinkish in color, because the granules that are present in the neutrophilic promyelocytes can attain, some granulocyte can attain blue color, whereas others attain red color. That's why the color is quite hybrid form of. Okay. Now from this promyelocytes, the other type of cell, that is myelocytes, has differentiated. The promyelocytes and the myelocytes are quite similar, but the main difference is that promyelocytes is that cell lineage that formed before the myelocytes. Okay, so this is, as I just told you, pro means primitive. So these are old old cells, and the myelocytes are newer one cells or some advanced cells. In this, we can see that there is a nucleus is having slight notch. Okay, this is not very constricted notch is present, but they are slightly notched in all the cases. Same in the eosinophilic myelocytes and also in the neutrophilic myelocytes. Now from the myelocyte, they have perfectly constricted nucleus. Okay, and the cells that are having perfectly constricted nucleus or perfectly U-shaped nucleus are known as band cells. Okay, so now from the basophilic myelocyte, basophilic band cells forms, and eosinophilic myelocytes generates the eosinophilic band cell, and from the neutrophilic myelocytes, neutrophilic band cells are formed. Okay, now from this band cell, there is again changes in the structure of the nucleus, and now they are taking their complete form. They form the multi-lobed nuclei. In the case of basophil, it is the U shape changed into S shape nucleus. Okay, and in the eosinophilic, from the eosinophilic band cell, there is a change from the U shape to the bilobed nucleus. This nucleus is bilobed here. And from the neutrophilic band cell, it's having pink color, as I just told you, and It's multi-lobed nucleus. Okay, it's multi-lobed. So this is the formation of granulocytes. It also comes under the process of leukopoiesis. Okay. Neutrophils. Among other granulocytes, neutrophils are the most abundant type. About 60 to 65% of the total leukocytes that circulating in blood are neutrophils. So they are about 60 to 65% of the total leukocytes that are present in bloodstream. The name derives from the stain loving neutrophils. Phils means loving and neutro, it's a kind of neutral. So it can be uh, acidic loving or basic loving. The name derives from the stain loving. Actually, neutrophils have neutral loving of basics as well as acid stains. That's why the color of the neutrophil granules is pink or purple, which can say that this is the combination of blue and red. The blue from the basic dye and red from the eosin or the acidic dye. Okay. The basophils, we just going to learn about the basophils and the eosinophils, where the basophil stains with the basic dyes, that's why appear blue, and eosinophils stains with acidic dye and appears bright red in colors. But neutrophils, as they are neutral loving of acidic as well as basic stains, so they are some granules attained the acidic dye, whereas some attained the what we can say basic dye. So their colors look like in midway between the acidic and the basic, or we can say the blue and the red. So they looks around pink or purplish in color. Okay. Now, neutrophils are professional phagocytic cells. Their size ranges from the 12 to 15 micrometer in diameter. Okay. The shape. The neutrophils are smaller than monocytes and they have segmented nucleus. You can see they are having segmented nucleus, and they may be trilobed, multilobed, and all the lobes are connected together by a thin chromatin fiber. Like you can see here, these are the thread-like structures which connected all these lobes. Okay, so these connected fibers are known as chromatin fibers. So there are two subpopulations of neutrophils. The first one is neutrophil killers and the second one is neutrophil cagers. The neutrophil resides in the bone marrow until maturity, but during infections, they comes out to the blood and reach to the infection sites. Okay, and as they received the appropriate signals from other types of immune cells, they do not return to the blood, but they turn into pus cells and die. So wherever you see the wounds which contain pus, it is basically the neutrophils. Okay. How neutrophil works? They are very smart cells and uses different strategies to attack a pathogens, and these strategies includes the phagocytosis, which means ingestion. The second one is degranulation, which means release of soluble antimicrobials, which including granule proteins. So as these are granulocytes, their granules contains different types of enzymes and proteins which degranulated and means released from the cells and can kill the foreign particles or pathogens. Okay, and the last one is the formation of nuts. Next stands for neutrophil extracellular traps. It's a kind of trap that's formed by the neutrophil cells which help in attacking and killing the microbes or the microorganism. Okay, so let's learn each of these strategies one by one. So as I just told you that neutrophils are professional phagocytic cells. They are masters in phagocytosis. They are masters in phagocytosis process. They rapidly engulfed the foreign invaders. This one is the foreign invaders, or we can say a bacteria. This one is a foreign invaders. Here I am using a bacteria which is coated with antibody or complement. So this bacteria is first get coated with antibodies or complement. Okay, so this is, and these antibodies comes to the neutrophils. As we know that neutrophils are having Fc receptors on their surface. So these are the Fc receptors. Once this antigen antibody complex binds to the Fc receptors, they engulf it and form phagosome. So as these combinations of the antigen antibody complex once binds to these Fc receptors, then there is a formation of phagosome. The lysosomal bodies, or we can say the granular content, fuses with the phagosome. Okay, these fuses with the phagosomes and form phagolysosomes, where pathogens get digested. It breaks down into smaller and simpler fragments. Neutrophils are known for their protein destroying and bactericidal properties. Neutrophils can stimulate monocytes and macrophage. So it can stimulate macrophages, which are big phagocytic cells, and monocytes as well. These phagocytic cells, the monocytes, are present in the bloodstream. Monocytes. By secreting some products, thereby enhance the process of phagocytosis. Their secretions also induce formation of reactive oxygen substances, or we can say compounds, that involved in intracellular killing. These are involved in intracellular killing. Now the second process is degranulation. It involves release of primary and secondary granules. So we know that this neutrophils contains different types of granules, and these granules are characterized as the primary and secondary granules. This neutrophils release its granules to destroy foreign pathogens. As you can see, this is the bacteria, and here this neutrophil is going to degranulate its granules to that side of the bacteria. Okay, so it prevents host tissue damage. Neutrophils have two types of granules, as I just told you. The primary, which are known as eosinophilic. Primary one is known as eosinophilic. Okay, granules that are found in immature neutrophils. So these eosinophilic granules are present in immature or young neutrophils, and the secondary, which are known as specific granules, which are found in mature or mature neutrophils. Okay, so primary granules. The primary granules contain cationic proteins. They contain cationic proteins and defensins, which are help in killing bacteria. So cationic proteins and defensins helps in killing bacteria. The next is proteolytic enzymes and cathepsin. So that break down the bacterial proteins. So the primary granules also contain proteolytic enzymes and cathepsin. Okay, which help in break down the bacterial proteins, and they also contains lysozymes, myeloperoxidase. Lysozymes is helpful in break down the bacterial cell wall, and the myeloperoxidase, it generates toxin bacteria which kill toxin bacteria killing substances. Okay, the neutrophils primary granule secretions also stimulate phagocytosis. So this type of degranulation, it also stimulates the process of phagocytosis. So this is all about the primary granules. Now the secondary granules or the specific granules, which contains compounds that help in the formation of toxic oxygen compounds. Okay, they help, the secondary granules help in the formation of toxic oxygen compounds. Lysozymes as well as lactoferrin. Okay, lactoferrin. So all these content of the granules secretes out and help in killing any kind of bacteria or foreign microorganism. Now next is netosis. Basically, netosis is net, which is abbreviated as neutrophil extracellular traps. Just like we use fish trap or net to catch fish, the neutrophils makes itself a trap to kill microorganisms or foreign pathogens. So basically net is comprises of web fibers that you can see here. These are the web fibers. Okay, these are the web fibers which is composed of chromatin and serine proteases that traps and kill foreign invaders extracellularly. So basically, if you can see, this is the bacteria, and this neutrophils is extending web-like structures from its nuclei, having histone, elastase and myeloperoxidases, and basically they helps in trapping the bacteria or microorganism and kill it extracellularly. Now eosinophils. These are granulated WBCs or leukocytes that are having bilobe nucleus. So you can see in this picture, this particular eosinophils contain a bilobe nucleus which is connected together by a thin chromatin fiber. Their cytoplasmic granules stained with eosin. Okay, that's why they appear red in color. It has a tendency of degranulations while they are in bloodstream, due to which granules number in eosinophils varies. So we can't calculate the number of the granules that are present in the eosinophils because it has a property to degranulate while moving in the blood. The eosinophils play a significant role in killing helminth parasites as their granules contains cathepsin toxic proteins. The most important thing is that their surface having Fc receptors that bind to IgE immunoglobulin or antibody. Immunoglobulin E antibody which complexed with antigen. So you can see in this picture these green structures are basically the Fc receptors. The Fc is the fragment, the constant part of the immunoglobulins which gonna bind with these receptors. That's why these are known as Fc receptors. These cells are professional antigen presenting cells and they co-regulate the functions of other immune cells like helper T cells, B cells, neutrophils, basophils and mast cells and also dendritic cells. The interleukin 5 or IL-5 is responsible for its growth and differentiation. So the IL-5 is responsible for its growth and differentiation which is produced by the helper T cells or the Th2 cell. They further involved in destructions of tumor cells and promote the repair of damaged tissues. Now, mechanism of parasites killing by neutrophils and eosinophils. So, as I just told you that IL-5, the interleukin 5 is responsible for the differentiation and growth of eosinophils. So, this structure is eosinophil, okay, and this is our neutrophils. Basically the their work are quite similar, so I am telling you in a same slide, in a single slide, how these neutrophils and eosinophils work together for killing of helminth parasites. So this is helminth parasites which are having antigens that are shown in red. These are antigens on its surface. Basically, the antigens are nothing but some kind of proteins that are detected by the antibodies. So, there is an interaction between the antibody. So, this is the antibody. In case of eosinophils, this is antibody immunoglobulin E, IgE, okay? Because the eosinophils are having receptors which are particular for the IgE. So they are having the Fc receptors that are binding for antibody IgE. Okay. So this is an interaction between the antibody IgE and antigen. They form the antigen antibody complex. So this one is the complex of antigen antibody. Then this particular IgE is bind with the Fc receptors. This one that's shown in pink color. This is the Fc receptors of eosinophils which induces as soon as this antigen antibody complex binds with the Fc receptors. Why this is called Fc receptor? Because this gonna bind with the Fc fragment, the constant fragment of the IgE, which we will look in our upcoming videos in the structure of immunoglobulin. So this particular part is known as the constant fragment of immunoglobulin. So this part is gonna bind with the Fc receptors. And as soon as this binding takes place, it causes degranulation. Degranulation means releasing of all these granules outside of the cell. So just like the eosinophils, there are the neutrophils to have Fc receptors, and these Fc receptors are for IgG. So eosinophils are having Fc receptors for IgE, whereas neutrophils are having Fc receptors for immunoglobulin G. So once this IgG antigen complex binds with the Fc receptors of neutrophils, it's to get degranulated. It also causes degranulations of neutrophils, where release of lysosomal or granular content like hydrogen peroxides H2O2 or nitric oxide NO, which causes lysis of this helminth parasites. So in case of eosinophils, their granules contains EDN. It means eosinophil derived neurotoxins, the EPO, the eosinophils peroxidase, MBP, the major basic proteins, and ECP, the eosinophil cationic proteins. These combinations of proteins that are present in their granules causes lysis of helminth parasites. So all these basically help in the killing of helminth parasites. Basophils. They are the least abundant WBCs in blood. Even less than 1% of all WBC contents that are present in our blood. They have the bilobed nuclei, but chromatins that connect these two lobes together are not as visible, so they look quite like S-shaped. Okay, so they are having S-shaped nuclei. They readily get stained with basic dyes, hence granules stained with dark blue color. So as its name indicates, basophils, they are loving of basic dyes. That's why their granules stain dark blue color. Basophils have receptors that can bind to IgE, IgG, histamine and complement. So you can see there are different types. I am showing you different types of receptors in different colors. Some are IgE, some are IgG, some are histamine receptors and some are complement receptors. There are numerous granules that are present in the cytoplasm of the basophil that causes sometimes conceal this particular nuclei. Okay. These granules contains histamine, peroxidase, platelet activating factors, heparin and lots more. Now this is mast cell. Mast cells are also known as mastocytes or labrocytes, which are present in the connective tissue. So this is the structure of the mast cell. Their cytoplasm contains granules which are rich in histamine and heparin. Okay, so both the mast cells and the basophils are quite similar in their structures and even the granule compositions. They are also developed from the myeloid stem cells, as we know, so like other granulocytes. That's why the mast cells is also considered a granulocytic cell. They play significant role in allergic reactions, defense against pathogens like parasites, and having protective role in wound healing process. They are also involved in angiogenesis, immune tolerance, and in inflammatory responses. Now, how basophils and mast cells work. So you can see this is B cell and this one is TH cell. As B cell their immunoglobulins, that are membrane bound immunoglobulins, as soon as they interact with the antigen, the B cell can activate it, and after phagocytosis they present the antigen and work as antigen presenting cell. That's why co-stimulate the T helper cell. So as soon as the B cell activation takes place, it differentiated into plasma cell. The plasma cell is going to produce the immunoglobulin E antibodies. So there are lots of production of the IgE antibodies. Okay, so after that, there is a binding of IgE with Fc receptors on the basophil and mast cells. So we know that basophil is present in blood and the mast cells are present in the connective tissues, and they also having the Fc receptors on their surface. So this IgE is gonna bind with the Fc receptors on the basophil inside blood, and the mast cell, the Fc receptors on the mast cell are going to bind to the IgE. Okay, now after that, now again, if an exposure of the allergen takes place, there is an activation of cells to release mediators, or they causes degranulation. So as soon as the same allergen that is present in the activating phase of the B cells, the same kind of antigen bind to the Fc receptors on the basophil and the mast cell, then this causes releasing of their mediator molecules. Okay, or we can say degranulation takes place, which causes release of their preformed mediators that are present in their granules. So from the basophils there are vasoactive amines and lipid mediators, and cytokines are also released from these basophils and mast cells. Basically, the amines like histamine and serotonin causes vasodilation and increased vascular permeability. The cell releases granules containing preformed mediators like histamine, which causes vasodilation and increased vascular permeability. They also release prostaglandin, leukotrienes, and other mediators, or we can say platelet activating factors and cytokines. So these are the different types of mediators that are secreted by the basophil and the mast cells, which gonna helpful in the inflammatory or the immune responses. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.